So, welcome to the next session, all awake after the coffee break, on cellular biology. So I'd like to introduce my colleague and first speaker, David O'Gorman, who is a research professor in the Department of Biochemistry at Western University in Ontario. And he has a research program on Dupuytren Disease, cell biology, and biochemistry since 2005 in the Rod McFarlane Hand and Upper Limb Center on controversies in cellular events.
Controversies indeed. Could I get my slides up here? There we go. Fantastic. Thank you very much, Elsa, for the introduction, and I have no disclosures to present. I thought we'd start the day with a very brief 101 on cell culture, because I appreciate that some people maybe you're not familiar with human cell culture, and this will lead into why this talk is entitled what it is, about controversies. So for the last 100 years or so, we've been getting progressively better and better at isolating pieces of tissue or organ from humans, taking them to the laboratory, growing out cells, and being able to study those cells in the lab. And when you do that, essentially one of three things happens. Either the cells will basically just fail to adapt and die, and that tells you that the culture conditions you're using simply aren't suitable to support the cells you're trying to study. Or they might attach to the substrate and temporarily adapt, but they typically won't replicate, in which case you might have, I don't know, a week or so to do some studies on them, but then basically the cells are useless. Then finally, you could have the potential where the cells might adapt and attached potentially to tissue culture. If they're adhesive cells, they may not be adhesive, and they may replicate, but when doing that, they typically retain some, but not all, of their original characteristics. And it is this that I think has risen the controversies associated with cell culture for Dupuytren disease, because I have heard from colleagues comments something along the lines of, culturing cells from patients with Dupuytren disease is an expensive waste of time. When you take the cells, you put them in the laboratory, they lose characteristics which are associated with the disease, they turn up other genes and things which are associated with adaptation. You can't tell the difference between an adaptive change and a disease-associated change. It's all very difficult, and so basically we shouldn't be worried about doing this. After all, we have genomics and epigenomics and metabolomics and all kinds of omics that we can do directly on patient tissues. Why would you bother to culture human cells? Is there anything we can actually learn from these things that you can't learn from doing basically all of those other omic studies? So I'm going to try and convince you that there are. And essentially, I'm going to start first with a quick overview of what cells you can actually find in a typical Dupuytren disease tissue. So this is some immunofluorescence and also some fluorescent staining of Dupuytren tissue. This is from Neil Henderson's laboratory. And if you so all these little blue dots here indicate the single nucleus of a cell. And if you look, there are a couple of little bits of yellow sort of staining here, and that indicates the presence of this marker, which tells us that this Dupuytren tissue has mononuclear phagocytes in it. These are cells like monocytes and macrophages. In addition, you can also look for a different stain. Again, blue dots are basically all of the nuclei of various cells in the tissue. And you can see that we have some endothelial cells derived from blood vessels. And we can also use a third marker, something like platelet-derived growth factor receptor beta. And as you can see, the very vast majority of the cells in this Dupuytren disease tissue fall into a third category, which we would refer to as mesenchymal stromal cells. These are cells, essentially various different kinds of fibroblast and myofibroblast. We don't really know how many there are, but there certainly would not want to give you the indication that this is only two types of cells. It could be 8, it could be a dozen. When you do cell culture, we see these loss of disease-associated characteristics. And rather than reflecting some failure of the cells to sort of replicate disease conditions, I would suggest it is in fact a failure of our culture systems. And that if we place these cells in conditions which actually mimic the disease, what I would refer to as a biomimetic. So we get away from the practice we've been doing for 50 years of putting cells on plastic dishes and instead start to come up with more complex culture systems. We can actually retain and regain most, if not all, of the disease characteristics for Dupuytren's disease. Now, in my experience, 3D matrix sort of based biomimetics under mechanical tension so that we get biomechanical inputs, also containing multiple cell types, are probably the best systems. However, you can learn fairly simple things doing fairly simple biomimetics. For example, we can ask questions like, do myofibroblasts in these cords actually remodel the collagen abnormally? So to answer that question, we thought, well, why not just grow some cords in the lab? So we did. So here we are growing some Dupuytren's disease cords in the lab. I'm using a system here which is very old, the flex cell system. It's a biomimetic system, been around for more than 50 years now. Without going into a lot of detail, there are little foam pads here, and stretched between them is a piece of polymerized collagen. And in this case, there's no cells in that, so it's just a piece of collagen. Over here, we've added some cells from the base of the aponeurosis of patients who are undergoing carpal tunnel release. These patients have no history of Dupuytren's disease. The last two sections over here are patients with Dupuytren's disease. The last one contains cells which are actually from a disease cord. And these cells here, which are particularly interesting, are derived from the fascia adjacent to a disease cord, but are not themselves visibly affected in any way. So as far as I can tell, at least from microscopy, they look just like normal fascia. And over time, if you grow them out, they contract, and you can develop little cords, just like these. And so some of the take-home messages here are normal cells can form a cord in the laboratory, and they have a general reduction of somewhere around 70% of their original volume up here. As you see, there is a modest reduction in the absence of cells, and that's basically just due to collagen polymerization. However, patients with Dupuytren's disease have a marked 10% increase in their ability to remodel collagen. So these cells are abnormal. They can remodel collagen abnormally compared to normal palmar fascia fibroblasts. If we do some staining to look at the cells, we can see that the cells will have some slightly different morphologies too. You can see these cells here are very stretched because these cells are actually under a lot of tension. These cells are much less stressed because these cords here are under substantially less tension. We can also do some staining to look at collagen remodeling using a system like this. And basically, it's something called picroserous staining, and it tells us something about collagen density. And as you can see, Dupuytren's disease cells are capable of remodeling this collagen very densely, less so for the adjacent prefibrotic cells, less so again for the normal cells. So we can learn these things simply by doing three-dimensional cultures, and these are relatively reproducible and simple. So if these mesenchymal stromal cells are all you need to make a cord, what are the phagocytic mononuclear cells doing? Well, maybe they're activating the fibroblasts and myofibroblasts. Or maybe the myofibroblasts are activating the monocytes and the macrophages. Or maybe both things are happening. So let's find out. And to find out, we could use another biomimetic, fairly simple biomimetic really, but takes a little bit of explanation. So I'll just go into that now. Essentially, this is just a 6-well plate. In the top here, we've taken some immortalized human macrophages and we've embedded them in little collagen spheroids. So they're bobbing around in the media in here. Macrophages, as you probably know, are adhesive cells, and so they have adhered to the collagen inside these little balls of collagen bobbing around in tissue culture media. Down the bottom down here, we've taken some myofibroblasts from Dupuytren's disease or normal tissues, and we've allowed them to contract that collagen and place it under tension. And so these cells have all developed into myofibroblasts. They're all very tensile. And basically what we can manage to do here is grow these cells in the same tissue culture media. So a few things to note. These cells can't touch these cells, or vice versa, but they can send each other biochemical signals. So secretions from these cells can be reached here, secretions from these cells can be reached here. And we can ask questions like, well, does it matter? Do the cells care if there are myofibroblasts in their immediate environment? And the answer is yes, they do. So if we look, for example, at gene expression, now we're going to focus on the gene expression here of the cells in the top, the THP1 macrophages, human macrophages. And I've outlined in red for you many of the inductions that we see in gene expression simply because they're in the presence, albeit they cannot touch these other cells. Some of these effects are general. All the controls and the disease cells have impacts. Some of them are more specific. We only have upregulation of interleukin-1 beta, for example, in this specific example. And so we can conclude that that these macrophages, in fact, do significantly increase the expression of some genes encoding inflammation-associated cytokines when they are co-cultured with myofibroblasts. Well, what about the reverse? Well, actually, before I get there, you might be wondering if gene expression leads to actual protein production. And in most of the expression analyses we've done, it does. So, for example, we see co-culture-induced upregulation of the interleukin-6 gene, and we see upregulated secretion of interleukin-6 into the media. What about the reverse? What happens to these myofibroblasts if there are macrophages in the neighbourhood? What happens is that they, and we are having going again from isolated culture, co-culture, isolated culture, co-culture, isolated culture, co-culture, left to right in all of these panels. As you can see, there are many significant increases in gene expression simply induced by the presence of macrophages in their environment, even though they can't touch them. And so, similarly, we can conclude that there are increases of expression of genes encoding inflammation-associated cytokines when cultured in the environment of THP1 macrophages. Now, and TNF expression, of course, is of interest to many people here. And so one of the things I thought I'd throw in here was the TNF sort of expression regulation in this system. If you look carefully, you can see that these cells in isolated culture, so in the absence of the other cell type, we see very low levels of TNF gene expression, although it is actually significantly increased in Dupuytren disease cells when in three-dimensional culture, not in two-dimensional culture. But when we look at these macrophages, they really don't do very much at all. However, if we look at them in co-culture, we not only get a dramatic increase in expression in the myofibroblasts, but not in the macrophages, and we see a dramatic increase in the secretion of tumor necrosis factor in the media, leading us to conclude that most of the TNF, at least in our model system, looks like it's coming not from the immune cells, but from the macrophages. So this would lead us to this synergistic interaction that we're talking about here. So essentially, macrophages are not driving the inflammatory milieu in Dupuytren disease, and myofibroblasts are not driving the inflammatory milieu in Dupuytren disease. It's the interaction between them that is driving the inflammatory milieu in Dupuytren disease. Now, I want to sort of finish on what I think is a very unusual and potentially predictive cell type. And this is what I refer to as these pre-fibrotic cells. Now, you remember I mentioned that these cells are fibroblasts or myofibroblasts, depending on how you culture them, and they are isolated from visibly unaffected palmar fascia, which is adjacent to a Dupuytren disease cord. So we would refer to these as syngeneic controls, meaning that they have the same genome. They carry all of the same SNPs and things that has been mentioned previously by Dominic and others. But these cells are, at the moment, not fibrotic. They appear visibly normal. If you compare them, however, at least at a gene expression level and also many other ways, the palmar fascia around these cells looks, as I say, very normal, and importantly, there is virtually no immune cell infiltrated in these tissues at all. There's just sparse fibroblasts. Yet, they do not display normal behaviours. These cells have marked changes in gene expression. They have changes in a whole variety of things, including beta-catenin association in genes. They have changed protein expression. They have all kinds of, as I think you saw, they have enhanced contractility in that early experiment, despite the fact that they're not actually derived from diseased tissue. So what does that tell us? Are these cells systemically activated? Perhaps having Dupuytren disease in the digit next door is sending something into the bloodstream, which is then activating these cells. Perhaps the SNPs and various pre-Dupuytren disease markers that they have inherited have already altered the protein expression and gene expression in these cells, even in the absence of disease. Perhaps, maybe it's both of those things. What I would suggest to you is that this suggests the existence of something that I would refer to as prodromal Dupuytren disease. So in other words, that it may be possible to detect Dupuytren disease in patients even before we see a nodule, because we are already seeing changes in gene expression and protein levels in visibly non-fibrotic fascia. So the take-home messages. Primary cells from Dupuytren disease can replicate many aspects of development if you culture them in the appropriate environment. Co-culture analysis tell us that it is synergistic interactions that promote the inflammatory milieu in Dupuytren disease and not specific cell types. There are particular roles for many other cell types. We really don't know what endothelial cells do. Prodromal Dupuytren disease, are there biomarkers of such things? Because if we can find a biomarker of prodromal Dupuytren disease, it may be possible to predict someone who is predisposed to getting Dupuytren disease before they have any symptoms, which of course would be of tremendous clinical importance. And lastly, a plea, we need much more realistic cell-based models. We already have lung on a chip, liver on a chip, heart on a chip. I don't know how many different things on a chip. We do not have a palm on a chip. We badly need a palm on a chip if we're going to move it further for new biomarker and new drug discovery. So finally, I didn't do any of this work, my students did. But I'd like to quickly point out Brianna Gonga-Carve, who did probably the lion's share of these experiments, and the rest of my lab who all contributed, and I'd be very happy to take some questions. Thank you.
Thank you, David, for this inspiring talk. Any questions from the audience, or remarks or suggestions? Not all at once. So, David, I told you this is going to be...
I think Paul has a question.
Why don't we have the palm on the chip?
That's an excellent question. I'd love to work with someone to... I've never been able to attract the funding to be able to generate one, but I mean, and that really is the problem. We have the technology. It's you know, even if we sort of lean on the shoulders of others, they say who generated the lung on a chip and the other things, it's all possible. That would, I think, have to be multicellular, but that also is achievable. So, yeah, it's a good question. I don't know why. We should have one.
So the students need to work even harder then.
True.
So, but if I understand correctly, at this stage, it's not possible to start testing the pharmaceuticals, the drugs on for instance,
I mean, right now in diseases like pulmonary fibrosis, for example, you can produce literally hundreds of lung chips. You can test 50, 100 drugs in quadruplicate all at the same time, you know, and have massive throughput and be able to come up with, you know, new and really interesting potential drug therapies. We can't do that for this disease, and there's really no reason why we can't.
Okay, so that's the next step. Thank you. Maybe we can...
Thank you.